Hello guys, hope I'm audible and visible. Can you see me? Can you hear me? If yes, say me a hi, hello. The first time I'm trying a smart board, so if there's any hiccups, sorry for that. Hello Layan, good evening, hope you're doing good. So hello Lalit, great. I hope I'm audible and visible, right? If so, say yes. So welcome back to everyone to PW Medit. I'm Dr. Ranjith and I'm going to start a new series called Robbins Read With Me. The main reason for starting the series is, I'll tell you what led to the series first. When I became an MBBS student, I had my seniors to guide. Most of the books what we take are the seniors books, that's all. I take a seniors book, the senior would already marked something like this, so it became easy for me to read a textbook. Now what happened is, when you read a real new textbook, these markings are not there. So the book looks like 1000 pages, very, very big, that to two sides like this, right? So it becomes very difficult. So what I'll do is, every single day, I'll come to the class, I'll make sure I'll read one tiny topic, 10-20 minutes max. Post that, I will also tell you how to take notes simultaneously from reading Robbins. So now in the class, listen to it, and after that, please go ahead and take notes. So 10-15 minutes, stay with me, 15-20 minutes, take a notes, so one topic is done. So by this way, if we start doing every alternate day, something like this, I am sure it will take some time for you to get into the group. But once you got into the group, it will be amazing. Lalit, my Hindi is thoda kharab hai, so my flow of teaching will be spoiled. That's why I am talking in English. If you have any question, you can type in Hindi. I can understand Hindi. Thoda thoda Hindi samjho hai. I'll try to talk in Hindi, but my flow of subject will be affected, right? Apologies for that. That's not my mother tongue. That's all fine. Okay. So if everyone is ready, so let's start. So today's topic is about cell division and cell proliferation. It's there in the first chapter of Robbins, which is generally not much concentrated on, fine. So now what we are going to do is, let's take this. I'll just zoom this here for you. This is there in the new edition of Robbins, the page number 38. So for cell division and cell proliferation, there is a definition in Robbins. Like if you can see here, I'll mark this for you guys. This what is the definition standing for, right? So I'm just going to take this part, nothing else, only till this part, okay? Till this to till this. So what cell division primarily says, or the function of cell division and cell proliferation, is very simple. It's going to help to maintain homeostasis. Can anyone tell me in the comment box what does homeostasis mean? You must have read that in physiology. Homeostasis means, come on, I am sure you guys know, do comment. Homeostasis is to maintain an equilibrium, that's all, right? So to maintain homeostasis, obviously I want cell division, because every day my skin is lost, so I have to replace that, right? That's the second part. For me to replace the dead cells and the damaged cells, I need cell division, right? That's for me definition. So this is there in Robbins. If you take any topic in Robbins, the first two lines is my definition. So if this comes in a 5-mark question in an exam, or you want to take a note, I just want you to take note like this. So what does exactly cell division means, right? Similio interior, great Tariq. So definition is, cell division, cycle and proliferation is to maintain homeostasis and also to make sure to replace the damaged and the dead cells. That's the purpose of cell division and cell proliferation, right? Amazing. So I know what is the reason now. Next, I need to know what are the sequence of cell cycle. I'll go to Robbins. Here, this part, the key elements. The key elements here, it's not required much because it's implied. Any doctor, once they start practicing after MBBS, they know that cell division requires nucleus, requires the cellular elements, requires the mitotic spindles. So this part, even if you ignore, I don't mind, because it has no implication in real life. It's there, which is self-implied, which every doctor knows. Ignore that part, right? So next part, if you come, I have highlighted something called a sequence of events, right? Hello, Sayank. So there's something called a sequence of events. In this sequence of events, where your cell cycle comes, like G0, G1, G2, M, and GS also, right? All the cell cycle event comes. So here, if you read, the sequence of events contains G1, G stands for gap in the cell thing, S for your synthesis, G2 for gap 2, and M for mitotic. These are the 4 primary things which happens in a cell cycle. In addition to this, I do have something called as an S phase, sorry, G0 phase. G0 phase is a resting phase in the cell cycle, right? You can see here, G0 or the gap 0 is a resting phase of cell cycle, which is a quiescent stage. I'm sure you must have heard about interphase, right? How many of you have learned the term interphase? Way back in 11th, 12th standard, right? Interphase is that the cell functions preparing itself for cell division. That might be your G0. So this entire thing is cell cycle. So when I'm going to take notes or I'm going to present in an exam, the main complaint what every senior of yours says is, if you read Robbins, you can't pass the exam. You can absolutely pass the exam. You can absolutely ace NEET PG. You can absolutely become an amazing doctor. So these are the information given in the next 4-5 lines. So when you're taking notes, just this, that's enough. The sequence of cell cycles was required. I have G0. I have made that sequence proper. G0 stands for your resting phase. After that comes your G1. I have written here also what is the function of G1. In Robbins, I'll show you where it is written. It's not written in this theoretical paragraph. So where the function of G0, G1, everything is written is go below. Robbins has amazing diagrams. That's where you have to utilize it. See, this is a cycle image where it is clearly written in G1, the function is nothing but to grow and have centrosome duplication. Can anyone tell me what does centrosome mean? Centrosome. Any idea? Centrosome is something which helps in your mitotic spindle formation. That also happens in your G1 phase. In S phase or the synthesis phase, it's self-implied, right? You will have chromosomal duplication, that's all, right? And then we have G2 phase. In G2 phase, primarily Robbins has not written anything in G2 phase. But what exactly happens in G2 phase, I'll tell you. And I want you also to just to envision, or I'm sure you know that. If not written in Robbins, you need not put it blank, right? What does M stand for? Can anyone comment on it? Great. What does M stand for? The mid part of the chromosome is centromere or centrosome? I've got few comments. What do you think is the mid part of the chromosome? It's a centromere, right? Centrosome is different from centromere. What does M stand for? Perfect. M stands for mitosis. So now tell me, what do you think will come here? Robbins has not given that. Doesn't mean that it's not required. M is for mitotic phase. So what do you think G2 will do? Can I say the cell should prepare for mitosis? Yes. Preparing for mitosis sense, should it make the mitotic spindles? You must have drawn in your school days these diagrams. The corner ones have to remove. That's what actually happens in M phase, right? So the preparation of mitotic spindles, the formation of mRNA, everything happens in the G2 phase, right? Perfect. Now here go to it. I wanted to create a note. G1 stands for growth in mass. The cell grows and also it duplicates your chromosomes. Then I have something called as checkpoint. I'll come back to this. What do I mean by this? Because I have just put a gap here. Because when I have a cell cycle, it's not just the phases, also the checkpoints are important for me, right? I have G1S, which is checkpoint. If possible, give a different color. Because here, it's a cell cycle. It's a break here. If possible, put a red color because break always is negative, right? So here, then S phase is synthesis. It's mean synthesis phase and chromosomal duplication. G2, like I said, it prepares for cell division. To be specific, it prepares for your mitotic spindle. The mitotic spindle formation, the centromere, everything is being done here. Again, I have a break, G2M, then I have M or mitotic phase, right? Perfect. Jonas, I'll try to do it as much as possible, right? Whenever time permits, I'm going to be here. And if you guys want anything in particular, put in the comment section or message me in the Telegram of PW group or my Instagram page. I'll definitely be able to take care of it, right? So the second part of the cell cycle is done, which is sequence of events. Now, in your biochemistry, have you read about rate limiting enzymes, rate limiting steps? How many of you have gone through that? Rate limiting steps of biochemistry, pathways. You remember them? Very important, right? Am I right in saying that? If you agree with me, say yes. Am I right in saying that every rate limiting step of biochemical pathways will have positive as well as negative factors which control the enzymes? Is that right? It will have? It will. So anything in my body should be having a very strict control if it's very definitive. So can I say this G1S, G2M, everything should be controlled? It has to be controlled, right? So every important step in my body will have both positive influence and negative influence. Whenever I need cell division, the positive influence comes to the top. Whenever I don't want cell division, the negative influence comes to the top. That comes in the next para of Robbins. If you look at the next paragraph of Robbins, I'll just go back, just give me a second. Go back to this paragraph. What this paragraph primarily talks is the cell cycle regulation, right? If you look at this, it's going to talk about the cell cycle regulation. The cell cycle is always regulated by both activators and inhibitors. Sayan, this is in the first chapter, page number 38. I think this is the online version. The page number 38 is where the topic is there, fine? So everything will have both activators and inhibitors. Activators is going to activate cell division, right? I'm just going to hide this. I want you to think and answer. It's a cell cycle dependent proteins. Do you know any name? Can you give any name for proteins which are involved in cell cycle? Anyone? Anyone? Comment on it. Yeah, thank you, Parth. Page 25 in hard copy. Can I call them cyclins? Yes. So these proteins are termed as cyclins, right? And I have to phosphorylate it in order to make it active and dephosphorylate it to make it inactive. If I need an enzyme which is to be phosphorylated, which group of enzyme in biochemistry have you read that the function is phosphorylase? Apart from phosphorylase, is there any other enzyme which can phosphorylate? Perfect. Cyclins and cyclin dependent kinases, right? Anywhere in my body, kinase phosphorylate. Apart from phosphorylase, kinase also phosphorylate. Amazing. That's what it happens here. There are both cyclin dependent kinases and cyclin enzyme which are associated. Cyclins are the enzymes. Cyclin dependent kinases are the activators, like a coenzyme or cofactor which activates them, right? So at every step of cell cycle, this is the one which is going to activate. So can I say there should be an inhibitor? Yes, there should be an inhibitor or not? I have to have an inhibitor, right? Robbins and medicine is very simple. When I have to have an inhibitor, I call it cyclin dependent kinase inhibitor. That's all, right? So the inhibitors here, though it's not written here, I'll have to go to the next page to go to inhibitors, but it's fine. It's very simple. It's not rocket science. It's very, very simple to remember and read. The only difficulty is in our head, right? So I'm going to call these inhibitors as cyclin dependent kinase inhibitors, CDKIs. But unfortunately, while naming them, some human being intervened and made a mess out of it. CDKI is the ideal name, cyclin dependent kinase inhibitor. But when you go to the family group of it, we call it CDKN, cyclin dependent kinase inhibitor. N stands for inhibitor, right? That's how we kind of classify them, right? So I know what does activate a cell cycle and what inhibits a cell cycle, right? And now this I'm going to take a note. When I take a note for this, sorry, I'm going to take a note like this. Give me a second. First time for smart board, so some amount of goof-ups will happen, right? Put a separate heading. Again, I'm repeating that the main concern for every student in Robbins is I'm not able to present a paper. It's simple. First was definition. Second heading was sequence of event. Third heading is cell cycle regulation. I have both activators and inhibitors. Both of them act on the cell cycle checkpoints primarily because that's where I can control, right? That's the checkpoints. I can control it, open it, close it, open it, close it, right? Then I have G1S checkpoint and G2M checkpoint. We wrote two checkpoints, right? G1 and S. The space between G1 and S is very clearly defined. G1 prepares for the synthesis. S doubles, right? What type of DNA duplication happens? You must have heard about something called semi-quantitative, right? Techno helper, I'll come to B53 soon. Give me just some time. You must have read about semi-quantitative method, right? In other words, I have two strands of DNA. It's both of them separate and they are exactly copied, right? By default, if I have an error in one copy, can I say the same error will be coming in daughter cells? Yes or no? Yes, I'll have it. So can I say before going to S phase, I have to stop the cell and look for errors. I have to look for errors. That's what happens in G1S. So G1S checks all the errors before synthesis. Because it's a normal cell, I go out in UV rays. Is there a possibility UV rays can cause mutation? It can. So it stops it, checks for errors, no errors, continues. In G2M, M is your mitotic phase. Now I am fastly producing ATCG as base pairs, complementary base pairs. Is there an error? Sometimes this complementary base pairs might not be right. Possible? If I don't check the error and I go for cell division, will the daughter cells have mitotic errors or mutations? It will have. That's what happens in G2M. G2M checks for same genetic mutations or errors before cell division, right? So I have two checkpoints. Both of their function is very, very clear here. It divides them, right? So if you want to give them in a better way or presentation, draw a diagram. Diagrams are always very well respected. Whatever I told, I have drawn in the form of diagram. Put a normal DNA. Put a DNA with a red color. I hope you can see the red color. That's an error in the DNA. Just some random image for it, right? Now I'm having a checkpoint inhibitor. Checkpoint inhibitor stops the cell cycle. Now again I'm going to come to you. Biochemistry will continue always. Throughout your life, biochemistry will not leave you, right? It's an amazing subject. We don't understand the importance of subject. Okay? So I have stopped a cell which has a mutation in G1S. Tell me what should be the outcome. Should I repair the error or should I kill the error or kill the cell? Comment what will you do? I am having a mutation in a cell. I have found out there is a mutation. I have stopped it. So what do I do? Should I repair or should I kill? Am I right in saying that I should repair? Amazing. Do you remember in biochemistry, you read about methyl mismatch repair, base excision repair, nucleotide excision repair? You must have read it, right? That's what happens here. It stops the cell cycle and tries for repair. If the repair is successful, the cell cycle continues automatically. If I am not able to repair it, let's assume I have done something, there's a mutation, I tried to repair, not able to repair. Should I kill the cell? Yes, no. Because if I don't kill the cell, the daughter cells will have mutation. Can I have the risk? I can't have the risk, right? If the repair is, I can't repair, if it's a failure, the cell cycle dies. Techno helper, you asked about P53. P53 is involved in the killing of it. That's one of the important thing, because P53 is one which tells, okay, it's in permanent inhibition. I'm going to check it. If it's not working, I will cause apoptosis. Apoptosis, cell death, which we'll read in the first chapter of Robbins, right? Perfect. So this is my entire goal of cell cycle regulation. So at the third heading, 5-mark question, right? 3 headings, you will get 3 marks. Our goal is to get 50%. You can get more than 50% by reading Robbins, right? Okay. We are all doctors, don't worry. You'll believe us when you come and sit in front of us, and we all amazing doctors will treat you, fine? Okay, now let's come to it. So I know what is the role of cell cycle control, right? Regulation. Now let's go to the control. So for control, before looking at this image, I want to explain what is a control is, and then we'll come to this image. I have activators and I have inhibitors as well. Activators is cyclin and cyclin dependent kinases, and inhibitors are cyclin dependent kinase inhibitors, right? So when we say activators, we already know that those are cyclins and cyclin dependent kinases, right? Those are them, simple. We have C, D, E, A, B. Remember, try to remember this order. I'll tell you why this order is important, right? Because in AIMS exam, there came a question that write the order of cyclins. It's a sequential arrangement MCQ. Again, all the MCQs in the exam comes from standard books only. You follow standard books, 100% you'll be able to ace the exam. Please believe me, there's no replacement to standard books, right? D, E, A and B, and D always pairs with 2, 1, 4 and 6. This controls the G1S phase. This is the most important one to control the G1S phase. To some extent, cyclin E and CDK2 also comes into play, fine? Apart from this, I have other 2 also. This is not exactly a cell cycle inhibitor because this doesn't attack the checkpoint. It helps to smoothly go to S phase and G2 phase. There's no checkpoint between S and G2. For a smooth transition, this helps, right? A and cyclin dependent kinase 2 and 1, and cyclin B with CDK1 helps in the G2M checkpoint, right? So I'm sure you now understand that the cyclins and cyclin dependent kinases works on the checkpoint primarily, right? Now let's come to the next. Next, your inhibitors. I'm going to divide inhibitors, and how Robbins divides inhibitors is this. Let's go back to Robbins here. This is where the inhibitors are there in Robbins. Look at this. Robbins calls them as 2 family of inhibitors. One family of CDKIs, which I'm going to call them as CDKN1. N stands for inhibitor. This is N1 family, and this is your N2 family. Fine. Just the way to remember, whichever is in N1 family, they are non-specific. They first formed one non-specific. And just a way to remember again, if it is N2, all of them will have number 1 in it. Okay? N2 has number 1, N1, the substances don't have number 1, right? N1 is non-specific, which means it can inhibit multiple cyclins, not just one cyclin, and it has P21, P27, P57. N2 is very, very specific. Which you think is the most important checkpoint? Let's see if some of you can comment on it. Which you think is the most important checkpoint? G1S or G2M? Where do you think error can happen more? Or where I need a checkpoint very importantly? G1S or G2M? Comment. Please do comment. Maximum you will make a mistake. You have 50% of chance of making it right. I just have 2 options. G1S or G2M? Okay, Parth has said G2M. Nayak has told G2M. Everyone goes going for G2M. Okay, great. I would like to differ a little bit. G1S is more important. I'll substantiate my claim of why I am saying G1S is more important, right? Because I want you to remember this when we come to neoplasia chapter again, fine? Let's say I am having a skin cell or any cell in my body. It's living for 10-20 days. It's becoming a little bit old. That's when it is going to divide, right? So when do you think error is more common? Like in the 10-day journey of a cell or during the cell division? In a 10-day journey, there is a possibility skin means some dirt can happen. I might have scratched it. Some infection could have happened. Mutation would have happened, right? There's a more possibility of having an error before entering the groove, right? So G1S is very, very important because pre-replicative errors are very much. If I correct them during replication, the errors are a little bit less. G2, I'm not saying G2M is not important. G2M is a little bit less. G1S is always more important, right? To give a real life analogy, you're trying to read and you have read. And revising, when do you think you'll make more mistakes? The initial time. When you try to read, there's a difficulty in understanding, there's more mistakes. When I try to divide, there's more mistakes. When I start dividing, the amount of mistakes is a little bit less, fine. So G1S is more important. So the second group is very, very specific, right? They inhibit your cyclin dependent kinase 4 and 6. This is with cyclin D, or this acts on G1S only, right? So here, this is the only information given for me in this, right? So how do I write this? So if I have to divide, I divide the inhibitors and I call them CDKN, cyclin dependent kinase inhibitor. Unfortunately, they have named it N. So I have two types, N1 and N2. In N1, it's multiple, it's non-specific. You can see here, it's multiple and non-specific. And everything which doesn't have 1 come into this. Can anyone comment on what does P stand in P21, 27, 57, 15, 16, 18, 19? What does P stand for? These are again very, very basic things. I'm sure quite a few of you know that. What does P stand for? P. P stands for? P is nothing but protein. A protein of 21 kilodalton weight is P21. A protein of 15 kilodalton weight is P15. That's all, right? P just stands for weight. There's no other information regarding P, just stands for weight. Okay, Yogesh, P is also for short arm of chromosome. When I talk about a chromosome, when I talk about any other protein, P stands for protein. So someone asked for P53. P53 is nothing but a protein with 53 kilodalton weight. That's all. There's no name to it, right? They have just named it based on the weight of the protein, right? So I have P21, 27 and 57 in the non-specific ones, and this specific one will be for G1S. And I have P15, 16, 17, 18 in that, right? Now this is an information. So if I write in the form of a paragraph, it becomes very difficult for an examiner to see. If I write in the form of this, it's decent for me. But let's go and write in the form of a circle image. It's much more convenient, right? G0, resting phase. G1, starting phase. I have a checkpoint G1S and I have checkpoint G2M. Again, use colors. Green for activators, red for inhibitors. In green, D plus 4 and 6 and E and 2 and A plus 2 and 1 for my this, and B1, cyclin B plus 1 for this checkpoint, right? And inhibitors, I have drawn one thing, that is CDK1 and CDK2, right? CDK1 is actually, this should be a little bit reverse, ignore this point. CDK1 is a very non-specific one which can inhibit this, this and this. But CDK2, which is a very specific one which can inhibit only my cyclin D with CDK4 and 6, right? That's very, very specific, right? This is a better way of representation. Now this is what actually is there in Robbins. How many of you feel that Robbins is very difficult to revise? Let's break that myth also. How many of you feel Robbins is very difficult to revise? So I've just taken half a page of Robbins, half to 1 page of Robbins, and we're just going to make sure it's very, very easy to revise. Comment if you feel it's difficult to revise. I also felt in second year it's difficult to revise. How many of you feel that? None of you? I'm very happy. Girish, in online the number is page number 40 to 38 to 40. If you feel it's difficult to revise, we read lots of things, right? Now let's go to it. Let's go to it. It's very difficult, right? Let's break the myth. See, this is what is there in 2 to 3 pages of Robbins. Now I'll show you just 2 images of Robbins. The first image is this. Can I say this has every sequence? Yes. It has every function of every sequence? Yes. And it also has the function of the checkpoints. The function of checkpoints. Yes. It has. This has the first half of notes what we took. You know the definition. You have the sequence of cell cycles here. You have the functions of the sequence, and you have the functions of the checkpoint. Done? That's the first image. Now let's go to the second image. When you go to the second image, this is the second image. In the second image, you have a few more informations. What cyclins are involved? What are the activators? And what are the CDKs involved? Again, what cyclin is involved and what CDK is involved? What cyclin is involved, what CDK? At exactly which point? That also is written. Not just that. Look at the top a little bit. The inhibitors. Look at this. The beauty of Robbins is everything is there already. This is your second group, or it is specific. If you look at this, 16, 17, 18, 19 blocks only cyclin D. But on the other hand, other three are very, very non-specific. The first group, this is non-specific, right? So can I say you can revise with these two images alone? Sure, Lalit, comment taken. I'll speak slowly. Is there a possibility to revise this with these two images alone? Yes, it is possible. So every textbook, what you read in your medical curriculum has a way. It will have 1 page or 1.5 page of theory. It will be converted into a flow chart or an image or a descriptive diagram. If you just read the diagram, it might become very difficult to revise. You read the theory, make a note like this by yourself, and then you revise the same Robbins in the form of a diagram. It's much, much easy. Which one you want me to repeat, Samantha? Okay, understood? So this is what I'm going to do with every Read With Me Robbins series. I'll take one tiny topic and I'll tell you exactly how to take notes for the topic. This is just a sample notes, and I'm 100% sure you guys have better handwriting and a better way of presentation than me, right? A definition, sequence of events, what is the function of the sequence, and your regulators, activators, inhibitors, where they act, and what is the function of the cell cycle checkpoint, and just a diagrammatic description of why there's a checkpoint. I have to repair or I have to kill the cell, and the control mechanisms of each and every checkpoint, okay, of each and every checkpoint, and inhibitors, the groups of inhibitors. Fine. Jane, last part, which part you want me to repeat? Okay, cell cycle controls. Okay, I'll go back to cell cycle. It's much comfortable for me to explain in the form of this image, right? For Jane and Samantha, just have a look at this. This is an image from Robbins which talks about entire cell cycle control. Fine. Okay, I'll go slow again. If you have any doubt, ping me. I have time. If you have time, let's learn together. That's all. I have a cell cycle. I have two checkpoints where I'm going to either close, check for errors. If there is error, I repair the error. If there's no error, I open. I let the cell go to the next phase. Right? So G1 to S, I have a cell cycle checkpoint. The guy who stops this, there's two groups of inhibitors. There's a very specific group of inhibitors called as CDKN2. That guy stops them. That's where your 15, 16, 18, 19 comes. They stop specifically G1S alone, right? I hope you understand this, okay? The next one, you have the non-specific one, CDK1A, where everything else is being affected. It's a very non-specific one. It can block any cyclin-dependent kinase, right? So in the G1S checkpoint, I have cyclin D with cyclin-dependent kinases and cyclin E as well. So in the GS to G2, I have cyclin A with cyclin-dependent kinase 2 and 1. The G2M checkpoint, I have cyclin B with cyclin-dependent kinase 1. So this is blocked only by one group of inhibitors. This is blocked by multiple group of inhibitors. Fine. Thank you, Rahman. Lalit, I am not from Kerala. I am from Tamil Nadu. I am from Chennai. That's why a little bit of inhibition in Hindi. I can understand Hindi, I can speak, but I am not able to teach in Hindi, right? I hope I'll learn soon. Maybe I'll teach you pathology, you guys teach me Hindi. Is that clear, Jane and Samantha? Great. So do one thing. I'm going to give you an exercise. If you have like half an hour of time, take the Robbins book, write the image, write a note of this cell cycle. It will take 5-10 minutes max. And tag me on Instagram. I'll tag you back. This is just a homework for you because I have to go back to school days so that we learn the way it has to be learned. Fine? Okay, great. So if it's possible, how many of you do think you will be able to create notes of this? I don't mind if it is beautiful or not beautiful. It should be productive, that's all. We'll have like this 30 minutes on alternate days. We'll cover a tiny topic. I want you to create notes and tag me on Instagram. Great. Genes, Lalit, I can't help. The way I look, I owe to my parents. Okay. Thank you, Priya. Okay, great. See you guys. See you in the next class. In the next class, I'll be discussing about the next topic to this, which is stem cells. I'll go one by one. I have time, good enough time. If you want me to take extra classes, do let me know. I'll definitely take extra classes. The goal is only one. The entire country should read Robbins. If you read Robbins, a selfish behavior. When you become amazing doctors, you're going to treat me when I become old, right? That's the only thing here, fine? Next topic will be on Wednesday, I think, and I'll be talking about the next part here, stem cells, fine? Alternative days, because I have a live session as well, I'll try to, if possible, if time permits, I'll try to take more as well, fine? Thank you. Thank you, everyone. See you soon in the next class. Bye-bye. And please don't forget to take notes and send it to me and tag me. Bye.